right, I think we'll get started. Welcome, everybody. I'd like to call the meeting to order. Welcome to the 1221st meeting of the Entomological Society of Washington. Today on May 6th, 2021. One note as we're getting started here, of course, this is being broadcast over Zoom. And please know that that we record these meetings and they'll be posted on our YouTube channel. And if you prefer to be anonymous, you can just not share your video and you'll remain completely anonymous, even though your name will show in our little meeting here. When it is posted on the recording, your name will not show, so you'll still be anonymous. And actually, I'll put a link to our YouTube channel in the chat here. So please visit. So the talks that have gone on for the past year are posted there. So you're welcome to go there and take a look at the talks that we've had already. Okay, first up I think I saw Gary Hevel. We'll have the reading of the minutes for the last meeting.
Okay, you can hear me now?
We can, thank you.
The 1220th regular meeting of the Entomological Society of Washington convened at 7 p.m. on the 1st of April, 2021. Jamie Zanizer presiding. As has been in recent months, the meeting was virtual through a Zoom format. There were 106 members and guests registered in attendance. Minutes of the previous meeting were read by recording secretary Gary Hevel, then approved by the viewers. Membership and Communications Secretary Elizabeth Young announced 3 new applicants for membership of the Society. Juana Maria Coronado Blanco, Ana Karen Serrano Dominguez, and Christopher Owen. President Zanizer encouraged new membership in the Society, citing details about the procedures. President Zanizer also mentioned that the guest speaker for the May meeting will be Marc Branham, a specialist on fireflies. For exhibits, Gary Hevel stood up to display his t-shirt that displayed 17-year cicadas from the 2004 emergence. David Firth followed this up by showing his t-shirt that showed a single individual of a 17-year cicada. Chris Owen introduced the speaker of the evening, Chris Simon from the University of Connecticut, whose title was 13 and 17-year cicadas, insects that count in fours. Dr. Simon presented a thorough review and discussion not only on the Magicicada species from the eastern United States, but fascinating facts about various cicadas from around the world. Her talk was generously supplemented with an array of informational slides. There were many questions from the audience after the presentation. The meeting was adjourned at 8:40 p.m.
Okay, thank you, Gary. Does anybody have any additions or notes to supplement the minutes? Okay, do we have a motion to approve the minutes as read?
Motion.
So moved.
And do we have a second?
Second.
Okay, all those in favor of approving the minutes, please say aye or raise your
Aye.
hand.
Aye. Aye.
Aye.
Aye. Any opposed? Okay, the minutes are approved as read. That's our makeshift gavel during these COVID times. Okay, next section will be reports from officers and committees. Do we have any officers that have anything to report? I assure you, we are hard at work, so we're working behind the scenes to keep things going. But nothing new to report right now. Introduction of new members and visitors. Do we have Elizabeth Young on tonight?
Hi there.
Hey, Elizabeth.
Yes, we have several new members this month. One moment, I'm sorry, I hear some shouting upstairs. I hope that isn't carried over. Okay, I think there's quiet. Great. Okay, our new members this month are Sabrina Hughes, Alina Avanisan, George Heimpel, Ken Schneider, M.E. Keogh, Judy Gallagher, Abigail Martins, Hazen Buckley, Jeff Clark, Michael Merchant, and Tara Preshnell. Our official members from last meeting are Juana Maria Coronado Blanco, Ana Karen Serrano Dominguez, and Christopher Owen.
Wow, that's excellent. Thank you, Elizabeth.
Yeah.
In the new members, I counted, I think, about a dozen.
Yes,
That's
we have
great.
11 new members. Yes.
Okay,
that's awesome.
great. Thank you. And do we have any guests here tonight that would like to introduce themselves? You don't have to, but if you'd like to, feel free.
Hi, Jamie Cromarty. I bring greetings from the American Entomological Society and want to say that we're really, really happy that we've been able to start sharing our Zoom meeting occasions with you folks and some of the other societies.
Great, good to hear from you, from one Jamie to another. Thank you.
Yeah.
All right.
Hello there. This is Andrew Lee. I'm a research entomologist with USDA in Beltsville. This first time I attend this meeting.
Thanks. Welcome, Andrew. Okay, great. Good to see everybody. And we'll move on to the next section, which is unfinished business. This is again where I give my little spiel about membership. If any guests are interested and are interested to know about becoming a member, you can visit our membership site, and I'll post a link to that in just a bit. Regular membership is $30 per year, or if you're a student, it's $15 per year, and that gives you access to our journal, the Proceedings of the Entomological Society of Washington, and also if you're interested in publishing in the proceedings, that gives you free page charges and up to 2 free color plates per publication. And you also would have the option to sign up for the paper copy of the proceedings, which would be an extra $30. And I'll post the link to that right now. Okay, on to new business. I have a little bit of new business. So as announced earlier in the year, the executive committee voted to reschedule our annual banquet. Our annual banquet is usually held in June here in the DC area.
That's just Lodores. That's normal.
Looks like we got an unmuted mic. But okay, maybe we'll push on. Okay, so normally the banquet is held in June, but again, due to COVID restrictions and uncertainties about gathering and so forth, we decided to postpone that until September of this year. And so right now that's scheduled for September 9th at Wood End Nature Sanctuary in the DC area. And so this actually is our last regular business meeting before the summer recess. So after this meeting, we'll be on recess until, well, we have the annual banquet, and then we'll begin regular meetings again in October. And so it looks like the next regular business meeting will be on October 7th. and we'll announce the speaker sometime later. Okay, on to the next section, which is presentation of notes and exhibition of specimens. So does anybody have anything special they want to show? Any specimens or books or images, hopefully relating to insects? Okay, no problem.
Yes, I do.
Oh, okay. Sounds like we have one.
Just trying to get the video up. This is Ed here. I don't know if I can get my video going. I'm trying. Start video. Yeah, I'm here. Okay, I want to bring this in, and it's a huge branch. I'm in a small room, but I don't know how well you can see this. I probably should turn it this other way around. This branch fell off. This is entomological. This branch fell off my neighbor's huge American beech in March during a windy day. And it has a huge cicada scar here. And I think it might be from 2004, because if you count out the nodes on the limb, so it goes out and it has 3 main branches, and one branch has, I think, 12 nodes, one has 15, and one has 17. So I think it might have been from the last big emergence. Also, there's some other cicada scars, which might be from the so-called annual cicadas. So it's a really cool limb. I felt very fortunate to find it before it fell apart. So I don't know what you all think. And then the other thing is, since we're in a big cicada, magic cicada year, I might mention there's a very humorous article in the Post today written by one of my colleagues' daughters about cicadas. You might want to look at that. You'll get a chuckle out of it, I think. That's it. Thanks.
Great, thanks, Ed. Very cool. Yeah, if any of you tuning in tonight are not in the DC area, we're bracing for the brood 10 emergence, which should be coming very, very soon. They're sort of burrowing up through their tunnels, and I've seen some nymphs. If you pull up some rocks, you can find the nymphs coming up these days. So it's going to be big. We're looking forward to it. Okay, so next up, I'll hand it over to Program Chair Al Norbaum to introduce tonight's featured speaker.
Okay, thanks, Jamie. Before we start, I just wanted to mention if anybody has suggested speakers for the fall meetings, please shoot me an email, or if any of you would like to volunteer to give a presentation, that would be welcome. Anyway, tonight we are very fortunate to have Dr. Marc Branham with us. He's going to be speaking about fireflies. Marc is an associate professor in the Department of Entomology and Nematology at the University of Florida, where he teaches several courses focusing on behavioral ecology and systematics of insects. His research focuses on insect systematics and the evolution of behavior. He's one of the leading authorities on fireflies and the evolution of their communication systems. Marc's research on fireflies has been highlighted in the traveling museum exhibit Creatures of Light, organized by the American Museum of Natural History, as well as documentaries by the BBC, NOVA, and National Geographic. He received his Ph.D. in entomology from Ohio State University and his MS in entomology and BS in organismal biology from the University of Kansas. In 2002, he was awarded the Theodore Roosevelt Postdoctoral Fellowship from the American Museum of Natural History, where he was a postdoc. And besides all that, Marc is just a really super nice guy, one of the nicest entomologists I know. So with all that, please take it away, Marc.
Ellen, thank you very much for that amazing introduction. Thank you. So I'm really happy to actually be here tonight. It's my first Zoom talk with this organization. I would much prefer to be in the actual museum on the mall with you guys, but you know, under the present circumstances, hey, that this is not at all bad. So for this talk tonight, I'm going to actually step through lots of chapters in my professional career that sort of highlight various questions that have been raised as I've been studying certain aspects of fireflies. And so if you will, this will be sort of like a walk at night through a forest as you see different attributes of firefly behavior. That certainly raises different questions. And my goal with this talk is to sort of document how my thinking has been changed over time as I've been studying fireflies, and then also how I've had to adopt several different skill sets to actually answer different questions. And so while I have done a lot of work within phylogenetic systematics, this talk tonight will only have a single phylogeny in it. And I know that that will be music to probably lots of people's ears, but I'm going to focus tonight especially on the evolution of these bioluminescent signals in fireflies. And so firefly flash patterns are probably the most famous, as you know, textbook examples of a species-specific courtship signal. And this illustration here is perhaps one of the more famous, which is by Dan Audie, which actually illustrates 9 separate firefly species, which are all flying around while they're flashing. And you will notice that each species has their own species-specific flash pattern. Some of them fly high, some of them fly low, some of them fly in the middle of this aerial space. And they also, firefly species will partition up the nights in separate acts of a play, if you will. And so a first one species comes out right around dusk and will start flashing for 17 minutes up to a few hours, and then that will then taper off as another firefly species comes onto stage. And so for quite a long time, fireflies have been the poster insect for a really discrete a very discrete species-specific flash pattern. And now, one of the things I certainly noticed early on, even as a young boy, is that if you watch this go on, all of these fireflies that are in flight while they flash within North America, these are all males. Okay, and then the females sit on very low-lying vegetation, and they wait until they see a male of their own species, which they find attractive, and then they will flash back at that male. He will flash back at her, she'll flash back at him, and they will have a short courtship dialogue until the male can actually home in on the exact position of this female. And it really doesn't take all that much time outside watching this to have the real impression that firefly females are quite choosy, and that males, as in most species, are not very choosy at all. And so one of the questions I had early on was, okay, so there's like all these different firefly flash patterns. That's cool. And that makes sense. But how do you get the evolution of these different flash patterns? How does one flash pattern turn into two? How does that diverge? Or like how does that like a gradually change over time into a completely different flash pattern? And so one of the selection pressures I hypothesize might be shaping this would be sexual selection, mainly female choice. And so for like part of my master's thesis, I wanted to like find a firefly species which was fairly close by, which would hover. Males would like hover while they flashed, so that like I would not have to like replicate so that I wouldn't have to like replicate flight patterns while I tried to replicate male flash patterns to show females to see what about male firefly flash patterns females found the most attractive. Because like I was reasoning if in like one species females had a certain preference for a certain flash pattern parameter in their males, that could help to like change how that male flash pattern evolved over time. And to then test that, I like essentially with a lot of help from some like a very, very talented engineers, we had like built this like artificial male firefly, which was composed of a computer, which like I could construct I could create firefly flash patterns on that like had been based on the actual natural signals, and then change certain parameters of that, and then like show that to the female as a result of like a powering a small light emitting diode light bulb. And then the female, if she liked it, she would flash back. If she really, really found it attractive, she would then flash back very often and then like very, very brightly. And so here are some terms I will be talking about as I discuss how I've examined each of these flash pattern parameters within a single species of a Photinus firefly. And so the first three parameters I was assessing was the actual color of light, and then also flash train rate, and then flash train duration. And I found females really didn't care about like any of these three parameters whatsoever. It could be a green light, it can be an orange light, it could be a yellow light. Females really had no preference there, or with like flash train rate and then also flash train duration. So then I had these four parameters, which were like integrally integrated with the actual flash pattern. Okay, and so then I was studying one firefly species in which males hovered while they flashed 5 to like 7 flashes very, very rapidly. And so I thought maybe one of these four signal parameters might be the ticket for like how like some of these males actually got a flash response and some didn't. And so what I did to actually tease apart these 4 intertwined variables was to hold one of these parameters constant, and then I changed one of the other ones. And then by necessity, the 2 others changed necessarily. Here's an example of that visually, where like I kept flash length constant, and then I changed flash rate, and then like inner flash interval changed as a result as the duty cycle. With this experimental design, I could hold each of these 4 parameters constant and then like look at at the overall effect on a female choice. And so, as you guys probably already know, firefly flash patterns are also temperature dependent, just like frog calls and also cricket chirps. And so on the hotter summer evenings, lots of organisms signal at a much higher rate. And so like fireflies that will like also flash multiple flashes will actually produce those at a much higher rate. And so I then selected 5 separate testing parameters at like a specific testing temperature. One of those was like at the mean for that parameter of that population. And then I had 2 more testing trials, one above and then one below that mean at that temperature within the natural variation of that population. And then I had 2 more testing trials, one above and then also one below the mean, however, outside of the like normal variation of the species at that temperature. And so for like these 4 parameters, I was running 5 separate testing trials. And so like here's an example here where like I would hold flash duration constant. Here's the actual distribution found in that population. And then here are my 5 testing trials. And then like when you hold flash duration constant, what you get, which I show here in terms of flash rate, which I have like graphed down here, but I could have also graphed one of these other 2 here. There was actual directional female preference. If you compare each of these 4 with the mean, these 2 on the end were actually significantly different. And so like what this then told me was that flash duration, when you hold that constant, that was not the key part. There was like still certainly something about male firefly flash patterns in the species, which these females were like really finding attractive at certain levels and then like unattractive at some other levels. And so then I then tested each of these other 3, and I found when you hold flash rate constants, you get pretty much a flat distribution here. And so in this species of firefly, Photinus consimilis, females like the males that could flash really fast. And they also like the flash patterns, which were so fast, they would never be seen in nature. And so there seemed to be some kind of a physiological speed limit past which males just could not flash any faster, even though females really preferred super high flashes outside of that natural variation. I sort of jumped the gun there. And so this got a lot of press when this came out because this was the like first ever demonstration of a sexual selection on a bioluminescent signal. And it was also about fireflies and about female choice, and of course courtship and sex. And so it got lots of attention. But of course, lots of media outlets don't like generally put up graphs and charts. They sort of make it simple. And so this entire study was like essentially portrayed as male fireflies being very provocative and like super interested in say courtship. while I know that all fireflies have more than just 2 pairs of limbs, I kind of approve of this vest in a way. And so then my next question was, okay, what else is going on here? What you can't see in this habitat with all this flashing going on is that there are some other fireflies there too, which are active, but they're not luminous in the adult stage. All fireflies have a luminous larvae, but they're not all luminous in the adult stage. And in those species, they will use pheromones in their courtship behavior to actually locate mates. And so, like, one of my early questions for my doctoral dissertation was like, what is the most ancestral condition within the family Lampyridae? Is it pheromones? Is it a bioluminescent signaling? Is it glowing? Is it flashing? Is it a combination of those things? What is going on? What is the actual sequence of these behavioral characters? And to then do that, you have to have a phylogeny. So I had to learn phylogenetic theory, and like with my former advisor, John Wenzel, we had constructed a phylogeny with which we could then address this hypothesis, and this was one of the like earliest phylogenies, which was actually produced with fireflies, and showed convincingly that the most ancestral condition within adults was the use of pheromones as their courtship signal. And then you found pheromones were like also used in conjunction with some bioluminescence in the adult stage. However, that is like almost always in terms of a glow, not a flash. And then in like several of the more derived clades within that family tree, then you had like multiple evolutions of a flashing behavior. And so like literally what you had on these fireflies was the like eyes getting bigger and the antennae getting to be a lot smaller as they started stepping through these 3 steps over the evolutionary tree. And then later, Gavin Martin, with like a whole lot more data, had like also confirmed that. And you see up here that the most basal firefly lineage was in fact non-luminous in the adult stage. And so does like anybody out there know what this is? That's right. This is a larval firefly, and all firefly larvae look something like this. Some are wider, some are more elongate, but they have this like general morphology, if you will. But they certainly all have the same light organ morphology. They have 2 small luminous spots on their like 8th abdominal segment. And for a long time, it was a huge mystery why firefly larvae would have bioluminescence, and then what function that could possibly serve. It's also interesting as you like walk around at night and you find these larvae, as you start stomping on the ground, they will glow much more often, and they'll glow much more brightly. And so it was hypothesized these guys probably use bioluminescence in this like immature stage, not as a courtship signal, obviously, because they're, of all things, larvae and they can't mate, but they are using bioluminescence as a aposematic warning signal. And we also see quite a bit of evidence for this in some adult fireflies as well. And so this is a firefly which is like generally crepuscular, and they use obviously pheromones because they have these like really, really fancy antennae. So they have like lots of surface area, lots of like sensory cells covering all of the surface area. But to me, after like having taught a lot of insect behavior, and then like also seeing a lot of organisms in the field, this sucker is clearly trying to, I don't know, communicate something. Either like that's an honest signal of like, hey, I don't taste good. Don't try to eat me, or like I'm a pretender. I kind of look gnarly, but I'm just really not that bad. I'm just faking it. And in like 1978, Tom Eisner and some colleagues isolated several different chemicals from fireflies, which were called lucibrophagens, that were like a similar in their chemical composition to the defensive chemicals found in toad parietal glands. And these can be found in defensively effective concentrations also. I can also tell you I have put fireflies in my mouth, and it's really not a very pleasant experience. And so they don't taste good, but I haven't stuck a toad in my mouth. I think I'll pass on that. But it's certainly interesting, and there's a lot of evidence that like most fireflies seem to be chemically defended, both in the immature stage and the adult stage as well. But there's a lot of species out there that haven't been tested for this specifically. And so this is a firefly that I tried to collect up in the cloud forest. Now that I can see some grins out there, I know now this is not a firefly, but when there was not a lot of light out and I was up in the cloud forest, this looked a hell of a lot like a firefly to me. And I tried to catch it, but it didn't move like a firefly. It moved like a roach. This is a cockroach that looks a lot like a firefly that is found in the same exact area. When you flip it over, it's got a light colored patch on the end of its abdomen where you might find a light organ if this were a firefly. And I put this one in here for my former student, Gino Nerns. This is a cerambycid beetle, which also looks a lot like a firefly. And then you flip it over and it has this light patch down on the end of its abdomen. And what is interesting here is if you really zoom in on that, that's actually created almost entirely by light colored seeding. And so one of the more difficult things here, if you want to study mimicry systems, which can be really tough sometimes, is that it can be hard to test what's the model, what's the mimic, because there's a lot that has to be considered in terms of what is an appropriate predator and how are those predators actually assessing prey, and will they be fooled or not. And so I am showing you this, I hope, as stacking evidence that fireflies are distasteful, and they try to advertise that to the extent that some other insects seem to also try to mimic them, either as Batesian or else Mullerian mimics. I'm not sure which, but I just hope to convince you as of now that fireflies do not taste good. And so I was then thinking, well, here's the situation where you have all these fireflies flying around, and they're certainly obvious when they're all lit up at night, because how much more obvious can you possibly get? They are lit up in these super dark environments. And if you watch fireflies flying around, they are not super agile flyers, but they fly with the nonchalance of a chemically defended insect. I mean, like, look at this. Look at this photo. These guys are like just like a cruising around on these warm summer nights, and they would be like easy pickings, wouldn't they? But I've spent a lot of evenings outside, and I've never seen a bat take a firefly. And I've often wondered, why is that? What is going on? Actually, could these courtship signals actually be warning signals? Could they both be functioning simultaneously in these two different roles? And there have been multiple studies looking at firefly aposematism and those predators which like might be feeding on them. And here are like some of those some of those represented here. However, these three studies looked at some terrestrial predators, and I was more specifically interested in these adult fireflies flying around that were super obvious in these environments, that were like apparently flying around without like any sort of danger at all. What was going on there? And there was one study done with bats and fireflies where like Moosman et al. Looked at like field observations of wild bats flying around in the same areas as quite a few fireflies. And they also looked at the attraction and the avoidance of some wild bats within flight enclosures and some bioluminescent lures. And they also looked at a whole bunch of bat guano to see if they could find any firefly parts in that guano. That's always fascinated me. I mean, you are a great insect morphologist if you can like pick through bat shit and be able to like ID what exactly those guys fed on. That is just really amazing. And they also did several different kinds of like palatability assessments, but this study primarily used wild caught bats, and they also used bioluminescent lures, which were not actual fireflies. And so I was always thinking, wouldn't it be really good if we could do some work with bats that like could be raised in captivity. And so like we could be sure with our experiments that like all of those bats in these study trials were completely naive predators. They had never seen a firefly. And so they were not coming into the trials having some preconceived bias based on their experience in nature a priori, right? And so like I was complaining about this to like one of my colleagues, Akito Kawahara, and like I was telling Akito, I was like, man, I know something about fireflies, but I don't know anything about bats. I gotta find somebody that is a like really good bat researcher who would be interested in like looking at the like firefly bat interface. And that's when Akito said, Mark, you need to meet Jesse Barber, who is at Boise State. He is the bat man. And so I then contacted Jesse and we talked about this, and like he was super, super excited. He had the bats and I had the fireflies down here in Florida. And so we actually devised this experiment where like we chose a firefly species that was found in the same exact location, in the same time of year, and was active at the same time of the evening as the species of bat we were going to test with. And of course, Jesse reared these bats. And so these bats in our trials had never seen a firefly until they were like flying in these enclosures in our experiments. And we used two control insects, palatable scarab beetles, and then like also palatable pyralid moths. And these two insects were like a roughly the same size as this firefly species, and they were not chemically defended. And so here's a short video of a simple palatability, a simple palatability experiment. And this like first part, we will like feed this bat a wax moth, which is apparently very delicious. And that'll be followed with a firefly, which is not so delicious. And so like watch this really, really closely. Here's the moth. And this is filmed in complete darkness, infrared video. Piece of cake. And then the firefly. This bat doesn't want any of that. Look at that. It's like feeding a small child broccoli, right? And then like, look at his mouth. He's like actually foaming at the mouth. Okay, one more time. And so we're pretty sure that our controls working pretty well, and our actual fireflies working pretty well too. Poor little bat. And then here were our two questions to be addressed. Okay. And so we set up this experimental design a lot like I set up the like earlier experimental design focused on like a sexual selection on a firefly flash patterns. This was set up so that like we could actually tease apart individual components of this system. Here's an example. So like first, we like had some trials with some fireflies, which were like a flying untethered in a room while they were flashing. And then we had fireflies which were flying and they couldn't flash. And then like non-flying flashing fireflies, and then non-flashing, non-flying fireflies. And we could prevent them from a flashing by actually painting over their light workings. And so here is some video of our control insect. Okay, untethered, and bat swoops down and just nails the scarab. Okay, and then the first encounter of this bat with a firefly, hits the firefly, grabs it, and then drops it. After a couple encounters, this bat flies really, really close and just goes right by the firefly. And so then what we found is that like when you run just the flying fireflies, which are not bioluminescent, which are not flashing, these bats can actually tell this is a firefly and they don't taste good. And they can tell this is a firefly as compared to like one of these controls, because they can learn this insect's sonar profile very well, okay? But when you have a firefly which is flying while it's also flashing, there is almost a 90% decrease in the probability of capture on the fourth night of testing. And so like bioluminescence enhances avoidance learning in this system. And so like essentially on the far right, this flying firefly is both being assessed by the bat in terms of its sonar profile sound, and then its visual signal, which is bioluminescence. Okay, and then if you compare fireflies which are like not flying and they're flashing with those that are flying and flashing, there is like almost a 70% decrease in the probability of capture. And so flight also enhances avoidance learning. So like if you think about, if you think back to that first slide of that like field of with those 9 firefly species, right? And they're all flashing different flash patterns, but they're all flashing different sorts of like aerial maneuvers. That also dovetails here too. And so the bats can see that, they can see the flash, and they can see how the flash moves through space, but they can also pick up the sonar profile as well. And so a bioluminescence, which is a visual signal, or flight, can be used to actually train bats not to feed on fireflies. But the combination of these two actually potentiated its effect. And this got a lot of press because this, let's see, this turned out to be the first example of a multisensory warning display which enhanced avoidance learning within a natural predator-prey system that had like never been demonstrated before. And so here you have like fireflies teaching bats with these two entirely different signals, with these two different signal modalities, vision and then hearing, those two enforce each other. And so what else is going on out there? There is like obviously deception. Every time I talk about fireflies, somebody asks me, yeah, and they're like, is that like one example, right, of that a femme fatale firefly, which will like eat the other fireflies? And that's true. So like in this environment of all these individual males flying around looking for a female of their own species, there are sometimes females of some other species which are aggressive mimics and will like mimic the like other species female. And here you have one of those aggressive mimic fireflies, which is a female Photurus, which is like feeding on like what is left of this Photinus male. Okay, and for a long time it was thought, well, so like this is one way female fireflies of like certain species in this Photurus genus can actually gain protein, but it has like subsequently been found by the Eisner lab that several Photurus species cannot manufacture their own defensive chemicals, and so they actually steal it from the other sympatric fireflies, which they feed on, and they then partition that to their eggs, and then their eggs and their larvae are just protected by those chemicals for a like very short period of time, and then the adults are not protected. And this was like made famous by my colleague Jim Lloyd back in 1969, and Jim unfortunately passed away this past year. And so this gets a lot of press because it's so Hollywood, and it's so amazing that there's lots of things going on out there at night that you might not know anything about. And so in conclusion, firefly flash patterns within Photinus consimilis seem to be under selection by a female choice, and that there is this like general trend from a more ancestral pheromone only mode of courtship to then pheromones that are like then used in conjunction with some glowing behavior, and then like pheromones drop out of the picture, and then the glows then like turn into flashes in like several different lineages. And bioluminescence and also flight cues within fireflies can enhance avoidance learning in bats, and then the combination of like light and also sound can also accelerate avoidance learning in these bats. And so here is a general hypothesis for how the evolution of firefly bioluminescence might have gone. Okay, where you have the first origin of light within larvae, and that was used primarily as a warning signal. And then you have the carryover of that light into the adult stage, probably through pedomorphosis, where it's then used in like both courtship and then also as a warning signal in these adults. And then further on up in this evolutionary tree, you like have the evolution of this predation using primarily mimic bioluminescent signals. And like what I find really interesting here is that like this serves as a wonderfully simple and then also charismatic example of how you can have the evolution of some very complicated systems through a small stepwise series of these additions over time. And then furthermore, you can have one trait evolve under like one function, such as like warning signals, which are under natural selection. And then further on in time, you can then have sexual selection act on that. And then that signal might be co-opted out of a warning signal into like both a warning signal context and a courtship signal context simultaneously. However, it can be really hard to figure out which of these came first. Was it aposematism in the adult or else courtship? And my colleague Gareth Powell has some like very good ideas about this, which was like he is working on, and so like I will not spill the beans on that. And so when you next see such an example of like outright amazing bioluminescence like this on a warm summer night, I wonder if you'll think about this as an incredible courtship display, or else as an incredible warning signal, or as a combination of the two. And so I just want to thank everybody here for their help and their suggestions. And I have a little bit of time, so I'm just gonna throw out several more tidbits as to like what we like currently working on. So like there are like multiple evolutions of flashing within fireflies. And there's a lot of different firefly light organ morphologies out there. And like one of my current PhD students, Kristen Dunn, has been CT scanning firefly light organs to try to get at how they're actually put together, how do they differ over these various lineages, and like how many novel components we actually might find across all of these different lineages. And so like this is a micro CT scan image, which is like a sliced in half. And you see this is dorsal up on top, and then this is ventral down here. And this wide band of tie-dye orange and yellow, that is this firefly's light organ. And like what is also really, really cool is that like this is so highly modified. This entire back layer here is a reflective layer. These guys actually have a reflective layer like your car headlights do to like push as many photons out of the front of the light organ as they can while they flash. And then also when you like a CT scan, you can also non-destructively sample what's going on inside of there. And you can like measure volumes and do like lots of cool stuff there. So this is to be continued. I just want to whet your appetite. And then I am like currently looking at some firefly fossils to see what sort of variation might be out there in deep time as far as light organs go. When did they like first evolve, and like are they big, are they small? What does that variation look like? And so hopefully we will have some more answers in this story going forward. Thank you very much.
Hi, Mark. I have two questions.
Sure.
One of them is the Photorus females. Those are the predatory ones, correct?
Right.
They must have two different flashing patterns, right? One to attract the wrong males and one to attract the right males.
And so so Photorus is a fairly medium-sized genus, and there's a lot of them that are in like Central and also South America. And so there's a lot we don't know about them, but the ones that are found inside North America, we still don't know much about them as well. Lots of those species, not all, there are probably 2 or 3 which have one single flash pattern, and those are not aggressive mimics. And then the other ones, which are aggressive mimics, they can have 2, they can have 3, they can have variable flash patterns. And so like one of the things that they can like make them so hard to study is that it can be hard to tell which of those like multiple flash patterns is their actual species specific flash pattern versus like other flash patterns that are also produced in some other behavioral contexts. And they'll also over like a 30-foot flight trajectory, they can switch back and forth. And so that is a genus which you don't want to try to study if you don't like really challenging biology.
Okay, thank you. My second question is, I wonder if you have some comments about the neotropical bioluminescent eleutherids. Are they mimics, or what's the function of that?
So like, yeah. So like that same, so like those bioluminescent click beetles also use bioluminescence for their pair formation and to find mates. And so like those systems haven't been very well studied, but in in general, you will find females that are more sedentary and it'll be glowing, and then the males fly around and they'll glow as they fly. But there's a lot that's a much more recent origin, I think, of bioluminescence in that lineage as opposed to fireflies. And I don't really know if it's just if it is a more simple courtship system, if you will, or that it's so complicated it just looks simple. If that like makes any sense.
Mark, I have a couple questions for you.
Sure.
Of the 410 morphological characters, and I understand you had other characters to produce your phylogeny, but of those morphological characters, were the majority of genitalic nature? And my second question is, if you were using genitalic characters, were they equally helpful in obtaining your phylogeny in luminescent species groups versus non-luminescent species groups?
Right. Excellent question. So like with that data set, there's like 410 morphological characters. Those were like general morphological characters that were like pulled from the entire body. And so like those were not like heavily sampled within one part of the firefly's body. And so and that analysis also was primarily focused on a family-wide level analysis. And so that did not involve all that many, if any, genitalic features, because like that like probably wouldn't be informative at that level. But there are quite a few firefly genera that can like only be differentiated on the basis of their like male genitalia.
Thanks.
Sure.
Mark, this is Al. First of all, thanks for the really fascinating talk. We have some also questions in the chat, so I can read them to you. Danny writes, in observing fireflies flying, I've noted that shining a bright light on them causes them to immediately dive for cover. This is similar to the way that many tymphanate insects respond to bats. Can you suggest what these fireflies are trying to avoid? And what do you think of the recent studies suggesting acoustic aposematism of fireflies in response to bats? I guess you covered that one. And he says, I've also photographed Photura species attacking and consuming moths. Is this common?
So yeah, okay. So let me see. So the first question, shining a bright light at fireflies as they fly. I don't know why they actually do that. I would hypothesize that you're just like overloading their sensory systems. But I don't know. I mean, it's like sort of hard to speculate as to like any other phenomenon that like might actually produce that, because fireflies are probably not going to encounter any other really, really bright lights in their environment naturally. So I'm sort of at a loss for, I mean, how to hypothesize what that might be due to. So the recent study of like a fireflies, I mean the acoustic aposematism, that was I think our study that you're referring to. And I don't know, Danny, I have never heard of Futurus attacking moths. And so like that's a new one on me. I mean, I've never heard about that. So like that is really, really cool. Tel Aviv University reported several species of fireflies also produce strong sounds, ultrasonic frequencies. Yeah, I don't know about that. I got to look that up. Very good.
Dave Firth says, a bit off subject of your talk, but is there evidence of firefly abundance reduction, AKA firefly apocalypse?
Hey, Dave. Yeah. So that's an excellent question. So like there have been quite a few papers that have been published recently, like in the past 5 or 6 years on dropping firefly numbers, and like lots of that seems to be due to either like habitat destruction or else like also light pollution. Lots of fireflies that are like employing bioluminescent signals at night to like actually find mates. If there's a lot of anthropogenic light in these habitats, lots of the fireflies either won't know that it's time to come out and start flashing within the window that species has been adapted to like a flash in, because it's just simply not dark enough. Or if they start flashing, it can be harder to see females on the ground because females have a much smaller light organ and they're not that they like don't produce as much light in their response as do males in theirs. And so certainly there is a pretty strong feeling out there that like light pollution has a like very serious negative effect on fireflies, and you guys see this all the time, right? As naturalists and as like entomologists out there, in today's culture, most people seem to be afraid of the dark. They just don't go outside at night when it's dark. They like have to like turn on all the lights. Even like neighborhoods have to be fully illuminated. Lots of people light up the outsides of their homes and their yards. And we know this has lots of like really, really negative effects on birds, sea turtles, lots of like nocturnal foraging organisms. And I just think it's so sad that like not only are we so afraid of the dark that we have to light it up, but like we are like trashing all of this biology happening out there because of our irrational fear of the dark.
Mark, Danny had a follow-up comment. It says, recently researchers from Tel Aviv University reported that several species of fireflies also produce strong sounds at ultrasonic frequencies during flight that can be recognized by bats. The authors hypothesize that this is a form of acoustic aposematism, warning bats to keep away. Is it unknown how common this feature might be among the many different species of fireflies?
So I am not familiar with this paper, and I don't have any experience with fireflies producing ultrasound. But the like funny thing about ultrasound, if you don't have a back detector, you can't hear it, right? So I mean, they like actually might be doing it, but like we just like haven't like thought about pointing a bat detector at flying fireflies to like see if they're actually producing ultrasound. And so like this this really sounds a lot like what like has been documented among like Euberiid moths and bat predators. Yeah, that's exciting. I gotta cool. Thanks for that link. Yeah, awesome.
Hi, Mark. Great talk. This is
Lourdes,
Lourdes.
thank you.
I was wondering if you could share with us your favorite resources on fireflies that we might be able to go. We get a lot of questions about fireflies, and we can share with our neighbors or any other people who are interested.
Right. Excellent question. So, there is a field guide that is available, and it's by Lynn Faust, and like that is on fireflies of North America, and she has pictures and she has distribution maps, and like that is the single most easiest to employ resources to try to find out what's happening in your yard, which species are you, if you like see fireflies flashing early in the evening in that area, are you also likely to like see some other species later on at night and things like that. And there is another book which is by Jim Lloyd, and like that was self-published, and I think that he sent a single copy perhaps to the Smithsonian's library, and he's also sent that to like several of the other libraries out there, and it's online too. I got to find that title here. And it's called Shadows. Okay, it's called Shadows, and it's by Jim Lloyd. And this is a book that is in like a PDF form online, and isn't like any other book that I've like ever read. This is something I think like everybody on this Zoom meeting would like find incredibly interesting. Jim Lloyd, I would describe as a like 18th century naturalist who was like he like had a keen eye, and he was always watching everything in nature, and he has like summarized all of his professional time in the field of working on fireflies, and it's like in this volume, and this volume is a quantification of like what he has discovered across multiple genera of fireflies, and it's like framed in this overall context of history and of philosophy and like literature, and he was just a scholar and a gentleman, and so like that is an absolute must read.
Excellent. Thank you very much.
Can I add a
Mark,
word?
somebody asked what was.
I would like to say that the Lloyd's is a naturalist long walk among shadows. That's the title of North American Photicus.
That's right.
And
that's right.
Yeah, the naturalist long walk among shadows. The second thing that I want to say is if you have male photinus, which you have collected night before, and one female who is virgin, and next morning you put that virgin with all the males, there will be huge commotion in that bottle. As one you have never seen in your life. So that means that all this flashing prowl is to bring the male closer. And I think, in close comment, it is the pheromones which do the mating, in my opinion. And it is difficult to separate the two.
Abner, how are you doing, man? That could very well be. It's good to see you.
Yeah, thank you. I'm just waiting to be 80 and 90 years old. I'm
You're in good shape.
Well, I don't know.
It's good to see you, my friend.
Good to see you too. And sorry about our friend Lloyd.
Yeah,
Jim. It was Jim who first showed me the difference between Photinus and Photurus, because the first time I recorded the eyes, the ERG from my samples was mixed, and he separated them.
Yeah.
So that's goes back to 1974.
Absolutely, absolutely. He was a master. Yeah.
Master, and and in my Photurus was the night species. For the Photinus, which I didn't know was the dusk active species, with yellow bioluminescence and very vision in yellow, narrow, made by colored filters in the eye. Photurus is the night active, no color filters, and the broad green vision. And they're all green in about 550 or 545 to 555 nanometers.
And so, guys,
you showed me.
I'm sorry, guys. If you like haven't met Dr. Lal, he's the expert on firefly vision and their like spectrum, all of that, and so he's done a lot of work on that over the years.
The other question I have, you said there's 603 emission of some firefly.
There's what? I'm sorry.
You said that I thought there was no more emission beyond 580 or 590 nanometers. You said that there is an emission in 608?
No, sir. No, sir. I was just like testing over that range with my LEDs. And the like thing about LEDs, as you know, is that like they can be very, very dirty in their like emittance spectrum.
Okay.
And so I could only get certain colored LEDs. And so like I tried to cover all of the fireflies known visual range with those LEDs.
I got it. Yeah.
And there's a link.
We have time for one. We have time for one last question, Mark. Ed Lasowski asked, he says, I don't think we have fireflies in the Pacific Northwest. Any theories why?
Yeah, so I get this question all the time from like the folks who are in California and who are like always lamenting that they don't have fireflies out in California. But it's really interesting. Actually, there are fireflies found in the extreme West. They are like just commonly not the kind of species which we see really, really commonly in the East, meaning they don't have a behavioral profile where they produce a flash signals. Lots of the fireflies found out west have a behavioral system where the like females sit on the substrate and they will glow on and off, and then the males fly around and they will find the females based on their glow, or their glow plus a pheromone. And so like people who are out like hiking around, very oftentimes don't see those glows because they're so faint that you like have to have dark adapted eyes. And there's not a lot of people that are comfortable hiking at night without a light on because you like fall over logs and barbed wire and things like that, or you like walk off a cliff, right? But there are fireflies on the West Coast, and they also extend up into Canada as well, but they just have a much different behavioral system, so that we don't seem to see a lot of flashers out there. There are some flashing populations in some like isolated parts of Montana and things like that. But in general, if you live out west, you are most likely to see flashing fireflies on the Pirates of the Caribbean ride at Disneyland.
Mark, I'm Bill Cooper. I'm out here. I'm here at the University of Florida over in environmental engineering, but I also do butterfly photography. So I'll look you up and send you an email. We can get together.
Cool, man. Cool. Yeah. Absolutely. Let's have lunch or something.
Yeah, I'd love to do it.
Cool.
Are you vaccinated?
Yes, of course.
Yeah, me too.
We're the only two in Florida.
That's right. I'll look you up. I'll get your email and send you an email.
Cool. Awesome. Awesome. All right.
Super. Loved your talk.
Oh, thank you. Thank you.
Mark, we had a total of 59 participants tonight. So if we were in our normal ESW meeting room, it would be more or less standing room only. So I think everybody
Cool.
really enjoyed your talk.
Cool.
And you can't hear us all clapping, but we're all very
You guys
appreciative of your talk.
You guys are all awesome. I just wish I was like there in person because I love DC and I love the Smithsonian, all of you guys, and I just like look out there and I see so many of my old friends and it is great, really great. Good to see everybody again.
All right, so let's turn it back over to Jamie.
Thanks again, Mark. That was awesome.
Thank you.
Everybody, thank you. Really appreciate it. That was fascinating. Thank you, everybody out there, for joining us tonight. Does anybody have any other final comments or questions before we adjourn? All right. Thank you again, everybody. Meeting's adjourned. Good night.
Bye. Bye, Mark. Nice to see you.
Thanks a lot.
Bye, Mark.
Mark, all right.
Great
Bye,
talk.
bye.
Everybody take care. Stay